So, hello everyone. In this video, we're going to talk about medulloblastoma. So, in our series of different tumors, we're going to talk about tumors, almost all the recognized tumors in this series. And so, this is especially for my students who have been studying medicine, and even for some students who have been quite indulged in their the studies of nursing or maybe other forms of medical studies. So let's get started. So medulloblastoma, this is quite interesting. So this is the tumor of the CNS tumor. So CNS means central nervous system tumor. And so this is a malignant embryonal tumor. So this is a really dangerous tumor, not tumor that's easily uh, not so easy to deal with tumor, and this especially occurs in children. The cases of this happening in adults have not been significant or maybe very rare, so we don't really diagnose these in adults. So this is exclusively in cerebellum, but we're going to talk cases where this may spread and metastasis may be seen, but exclusively it starts in the cerebellum. So this is a grade 4 tumor. So in our previous video, we actually talked about the classification of tumor based on their different features and their ability to, cause death. So these are grade 4 tumors, the most dangerous kind of CNS tumors, okay? Tumors occurring in the brain. These are highly invasive. But one thing to be noted, these are very sensitive to radiation, so chances of curing are quite high, despite we dealing with a lot of problems. So generally, so this is the section of the brain where these are occurring, the medulloblastoma occur. So these are the brain tumors. So now actually let's move on to one of the other features. So normally these are just tumors, but other conditions that it may result is so rapid growth of the cells may occlude the flow of CSF since it is developing in the cerebellum. So what actually happens is there are the ventricles, and ventricles may be occluded by the rapid growth, especially the fourth the lateral and the fourth ventricle occlusion is commonly seen, the lateral ventricles, and leading to hydrocephalus. Hydrocephalus is basically, there are two kinds of hydrocephalus, communicating and non-communicating. In this case, what actually happens is non-communicating hydrocephalus may occur. So now let's move on to the pathogenesis. What are the factors that are responsible for the occurrence of cancer or medulloblastoma? So basically the most common pathogenesis that we have actually diagnosed is there are signaling pathways involved. So alternation in the signaling pathways involved in normal cerebellar development. So these are embryonal tumors, okay? Don't forget embryonal tumors seen in children. So especially the development of cerebellum is quite, hindered due to these the abnormal or alterations or the mutation in these pathways, genes that help these pathways. So the first one is so basically there are two pathways that are recognized till that. So the first one is the sonic hedgehog patched pathway. The other one is WNT beta catenin signaling pathway. So these are both signaling pathways. So this sonic hedgehog patched pathway, so this is involved in the control of normal proliferation of the cerebellar granule cells. So these granule cells, these are found in the granular layer of cerebellum. So basically involved in this cerebellar circuitry. So when we're talking about the physiology of the cerebellum, we're going to talk about the granule cells, but this is especially, so let's not dive into details. So the granule cells of cerebellum are not actually able to proliferate if this pathway is hindered, or there may be over proliferation. So basically there's this problem. So location, the insonic hedgehog patched pathway, the problem is generally seen in the vermis and the lateral column. The vermis and the lateral column. So when we're functionally dividing this cerebellum, we divide in the vermis, the intermediate column, and the lateral column. So basically this and this area, so this is the intermediate column, not really involved in the sonic hedgehog patched pathway. And there's this double anti-beta catenin signaling pathway. So basically, if there's certain mutations or hindrance of this pathway, we can see abnormal development of the cells in the anterior fourth ventricle or middle cerebellar peduncle. So basically, that's all. So middle cerebellar peduncle basically means in the area region in the pons, and the medulla lies in the inferior cerebellar peduncle. So the fourth ventricle, basically we have to understand the anatomy of cerebellum and the brainstem in order to understand this. But now our concern is tumors, so we're going to talk about tumor itself and not the anatomy and the physiology. So let's dive into the morphology, the most important aspect. Okay, so grossly it is well circumscribed, unlike anaplastic cancers, and it's gray in color when you're looking at it grossly, and friable, and may extend to the surface of cerebellar folia and involve the leptomeninges. Leptomeninges are arachnoid and pyomatter, and microscopically densely cellular with sheets of small primitive appearing. So this is highly cellular neoplasia. So densely cellular, high cells. So which means you can, there is mitotic activity, high mitotic activity, and sheets of small primitive appearing cells. Though there is high primitive, high cellular activity and mitosis, still the cells are small primitive appearing cells. Individual cells have scant cytoplasm, so when there is less cytoplasm, eosinophilia is decreased, eosinophilic features are decreased, and hyperchromaticity or basophilicity is increased in the cells. So you have to understand this. So we don't have cytoplasm, so eosinophilic staining is rather not seen, whereas we see the hematoxylin stain it. Okay, so hyperchromatic nuclei, less cytoplasm, and there is abundant mitosis. As I've seen, these mitosis means mitotic bodies, which is the replicating bodies, cell replication, or as the dividing bodies, and markers of cellular proliferation. So such as Ki67 are positive Ki67. So basically, if you take some of the cells, you can see that these cells are, if you check these markers, you can see them positive, which means these cells are actually dividing rapidly. They're actually undergoing cell division. And one other thing is hyperchromatic nuclei, and the other feature of the nuclei is they're often elongated and crescent shaped. Crescent shaped, these kind of nuclei. Okay, basically what it looks like is when you're in the, okay, so this shaped, elongated and crescent shaped, moon shaped. Okay, so basically this is the one of the most beautiful images that I got. So elongated and moon shaped, small blue round cell tumors, primitive celling, and there are 3 subtypes. So this is easy to understand. So there is the classical variant and there is the desmoplastic variant. Desmoplastic suggests there is more collagen. Okay, and there's large anaplastic. This is quite ridiculously dangerous. Though the whole tumors are dangerous, this one is the most dangerous one, anaplastic variation. So formation of homocyte rosettes. So homocyte rosettes, as I've explained in other videos also. So these are the cells, and there's this area which is not cellular. This is non-cellular area, and this is called neuropil. Okay, neuropil, and it contains fibrillar substances. Okay, and that's all. So homohydrosis and express formation of homohydrosis often express neuronal markers such as synaptophysin, and GFAP is less positive. So in this case, GFAP is lesser positive, unlike other tumors, and synaptophysin may be shown by this classic variant, this tells me so. And desmoplastic, the nodular variant, characterized by internodular areas of stromal response. So there are nodules, and there's this internodular space, internodular space, and another nodule, and this is the internodular space. And stromal response can be seen marked by collagen and reticulin deposition. So whenever you're asked what happens in desmoplastic variants of most of the tumors, there's this deposition of the collagen and reticulin in this, the actual cytoplasm, and there are pale nodules that have more neuropil and show greater neuronal differentiation. And a large cellular anaplastic variant, so this is large and vesicular nuclei, large nuclei, big nuclei, big nuclei, big nuclei, and there are these actually prominent vesicular nuclei and prominent nucleoli and cell wrapping. There are some structures that so like another cell in a cell, one cell, another cell together, two cells wrapping. So this is called cell wrapping. A cell in a cell. It seems like there is another cell inside the cell, and high mitotic and apoptotic indexes. So, and that's all. So anaplastic, high apoptosis, and mitotic indexes. But one thing you have to understand. So this is the end part. Remember, it has a tendency, okay? So in tendency to spread to subarachnoid space, and dissemination through CSF is common complication, giving rise to either extensive growth along the surface, like into icing, or nodular drop metastasis. So drop metastasis basically. So there's no, hematogenous spread or lymphatic spread, but actually the spread from the metastasis from the CSF, which tend to involve the cauda equina, which is the lowermost region of the spinal cord. So in some cases we may see this in spinal cord if the dissemination through the CSF is done. So I hope you understood medulloblastoma. I hope you liked my lecture. Thank you for your time, everybody. I hope we can meet in another series of tumors.